as chair of the Clinical Working Party, I wanted to welcome all the attendees for attending this fantastic grand round. And I personally thank Anne Puel, who organized and recruited fantastic speakers for today. I take the advantage of this grand round to inform you that there is now the Journal of Human Immunity, that is the new ESID journal. So I really encourage all the members of ESID to submit publications to this journal, as well as to review in case papers that can come to your email. So this is a good opportunity to launch the journal and to promote the scientific activities of ESID. So with this, I leave the podium to Anne and all the speakers. Thank you.
So hello, everyone. It's my great pleasure to welcome you to this ESID Grand Round session on autoantibodies against cytokine. So my name is Anne Puel, and I serve as the INSERM Research Director. I'm a group leader on the working on the genetic and the immunological determinant of severe fungal disease in humans. And together with Paul Bastard, we are leading a group working on the cause and consequences of autoantibodies against cytokine. So this within the lab of human genetics of infectious disease at the Necker Hospital Imaging Institute in Paris, and at the Necker at the Rockefeller University in New York. So as you will see, we have a very exciting program ahead with 5 internationally recognized speakers who will talk about 4 topics. And the schedule is pretty tight, but I really strongly encourage you to enrich the discussion by asking questions in the Q&A tab that we will address at the end of the session. So our first speaker, Dr. Hambleton, is a PhD and an assistant research fellow at the Center for Molecular and Clinical Immunology in Changchun University in Taiwan. So Dr. Hambleton has a special interest and expertise on autoantibodies against interferon gamma, underlying in particular, but not only mycobacterial diseases, and he has also a strong expertise in cellular therapy. He has also contributed to a number of work performed in the Dr. Cheng Lu School's lab on the role of neutralizing autoantibodies against GM-CSF in nocardiosis and cryptococcosis. So Dr. Hambleton will be presenting on autoantibodies against interferon gamma, their pathogenic role, and the therapeutic solution. Please, Hambleton.
Okay. Thank you. Thank you, Professor Anne. And in the beginning, I would like to thank about the organization to invite me to have this presentation today about the autoantibodies to interferon gamma. And the importance of the interferon gamma immunity in humans had been demonstrated in patients with inborn error of immunity. About back to 30 years ago, there is a rare case of children suffer from recurrent mycobacterial infection. And at that time, Professor Casanova had identified a rare gene called interferon gamma receptor 1, and find out this mutant will cause the unresponsive to interferon gamma and cause it to the recurrent mycobacterial infection in these children. And in the following years, there's several genes had been identified. Their group had uncovered the interferon gamma, and the IL-12 pathway will control mycobacterial infection in humans. In adult patients, there is a similar phenocopy in there, and they identify there's no underlying genetic defect. However, they have the autoantibody against interferon gamma, and they prove they have the inhibitory function. And the interesting thing is that this patient all comes from Southeast Asia. So when after training by the Professor Casanova, my mentor, Professor Zhenren Gu, have come back to Taiwan and want to recruit this patient. So we set up a simple in vitro activation assay. We use the whole blood sample and the PBMC to activate the patient, and you can see obviously the whole blood sample from the disseminated NTM patient, there's no detectable interferon gamma. And you can see the PBNC can produce interferon gamma in a normal way. Further, we find out we use the competition ELISA and the indirect ELISA to confirm the factor in the plasma has the autoantibody against interferon gamma, and they have a neutralizing effect. However, our next question is wondering how do these autoantibody perform their neutralizing? To address this question, we establish a single human B-cell cloning platform. We isolate the auto-react B-cell against interferon gamma from the patient PBMC through the flow cytometry. So we can reconstitute this B-cell into their heavy chain and the light chain sequence and produce the recombinant monoclonal antibodies for our further studies. So we tested 10 patients, and we had isolate over 100 monoclonal antibody from 3 patients, and we screened this antibody with the interferon gamma ELISA, and find out there's 19 monoclonal antibody against interferon gamma. We further use the mutant interferon gamma and some biochemical assay to find out these antibodies are categorized into 3 binding groups. We further use the in vitro functional assay and find out this antibody have different level of the neutralizing effect. So the interferon gamma is a cytokine, so it will bind to the receptor to transduce the signaling. So we are wondering, we perform in vitro interferon gamma R1 ELISA, and we measure the interferon gamma binding signaling on the R1 protein in the presence of the different antibodies. As you can see, we test several antibodies. The Cy1 antibody from the E1 from the Cy1 antibody, it can directly and reduce the binding signaling of the interferon gamma compared to the control or other group. And there is no detectable IgG amount on this ELISA. So we further move to a cell-based assay and confirm the E1 have no detectable IgG on the target cells. So here we can realize that and conclude that the E1 antibody can directly inhibit receptor interferon gamma, prevent interferon gamma binding to its receptor, and provide the neutralizing effect. But how about the other neutralizing antibody? The Professor Pierre from Germany, he had performed a single molecular tracking experiment to answer the interferon gamma binding to the receptor will perform the receptor dimerization. So we collaborated with the Dr. Pierre, and we hypothesized that maybe these other 2 antibodies will perform the neutralizing effect through the interrupt the receptor assembling. So as you can see here, we compare to the IgG control or the non-neutralizing antibody from these 2 group antibody. The 2B6 or 1EA, it can inhibit the R1, R2 receptor dimerization. So here only we can realize that there's another neutralizing mechanism. You will interrupt the receptor assembling of an interferon gamma binding. Additionally, we are wondering how about the role of the FC region of these antibodies. So we perform and establish a simple assay called the ADCC reporter assay. We screen all these 19 monoclonal antibodies with different level of interferon gamma. Obviously, you can see the antibody against Cy1 have a dose-dependent manner of the induce the FC receptor activation. We further confirm this finding with the LALA mutation antibody. The mutation is on the FC region, and it will abolish the FC receptor. So you can see here the LALA antibody obviously can reduce the FC receptor activation. So we further isolate the autologous monocytes and the NK cell to perform the several cytotoxicity assay. Compared to the control or the LALA antibody, only the Cy3 antibody can induce the cellular cytotoxicity. And we move the assay to the plasma, and you can see obviously some patient plasma still can induce the cellular cytotoxicity. So here we can realize that, and we will make a brief and stop summary. So through the B-cell cloning platform, we isolate the fully monoclonal antibody from the human, and then we find out if the antibody against the Cy1, it will prevent interferon gamma binding to its receptor. And however, if the antibody against the Cy2 or Cy3, it will interrupt the receptor assembly. Additionally, the Cy3 antibody can induce the ADCC. It could be another biological activity for the autoantibodies to interferon gammas. So, adaptive T-cell therapy has worked well for B-cell malignancy and is now being used to help with autoimmune disease. The chimeric autoantibody receptor T-cells are an upgrade CAR design that target autoreactive B-cells without compromising humoral immunity. So it was initially reported to treat an autoimmune blister disease caused by autoantibodies targeting the keratinocyte adhesion protein desmoglein 3. And the CAR adopt the autoantigen as the bed and recognize the B cell receptor and direct T cell to kill autoreactive B lymphocyte. As you can see, the desmoglein CAR T cell shows strong efficacy in eliminating the autoantibody producing B cell in vitro and in vivo, and it can effectively reduce the symptom of the blistering in an animal model. So, my talent colleague, Zhan Jie, who want to deploy this strategy for treating disease induced by autoantibodies to interferon gamma, and there's a challenge of design a bed to mimic cytokine without triggering their biological function. To overcome this limitation, we therefore design a mutant interferon gamma as a bed to develop interferon gamma CAR T cell for targeting autoreactive B cell against interferon gamma. Since the alanine and the histidine are essential for the interferon gamma receptor binding and the signal transduction, we substitute both residue with glutamine and aspartate to prevent interferon gamma receptor cross-reactivity. These two-point mutation on interferon gamma complete abrogate the interferon gamma signaling by the HeLa gas luciferase reporter system. And this is the prototype of our CAR design. We next introduced the interferon gamma CAR to primary human T cell by lentivirus and examine its surface expression. Based on the standing of MIC-TAC in the N-terminal of CAR, and surprisingly, we barely detect surface interferon gamma CAR in human T cell transduced with the full length of interferon gamma. We want to improve the membrane presentation of the CAR. We design several targeting interferon gamma variants as a CAR based on the identical epitope from the monoclonal antibodies. And to make our story short, we find out these two versions of interferon gamma variants as a CAR can be detected on the T cell surface, and they can against a different site of autoantibodies. And the next, we test the cytotoxicity of this CAR, interferon gamma CAR, against the leukemia cell that express the autoantibody BCR and the CD19 antigen. So compared to the positive control, the CD19 CAR, as expected, the C-terminal extended interferon gamma CAR show a broad cytotoxicity on target cell expressing the anti-interferon gamma autoantibody BCR. However, the AF version can efficiently kill target cell with the BCR against the Cy2 and the Cy3 interferon gamma. Altogether, our result reveal that the truncated interferon gamma can be used for CAR T cell against B cell expressing autoantibody BCR. And we find out the soluble autoantibody can bind to interferon gamma CAR T cell that might induce unexpected reaction and block the interaction with autoreactive B cells. We therefore evaluate the impact of the soluble antibody on the specificity and the potency of interferon gamma CAR T cell by in vitro selective killing assay. We mix wild-type cells and the target cell with specific BCR and co-culture with the CAR T cell in the presence of plasma. Compared to the wild-type cells, our results show that both interferon gamma CAR and the CD19 CAR T cell efficiently kill target cells expressing autoantibody BCR. Meanwhile, we observe a trend, but not significant. The plasma from the autoantibody patient decreased the cytotoxicity of interferon gamma CAR T cell. Overall, the soluble autoantibody do not compromise the specificity of the interferon gamma CAR T cell, but could potentially reduce their efficacy. We then assess the efficacy of interferon gamma CAR T cell to deplete target cell in vivo by adopting NAPSIS xenograft model in NSG mice. We established NAPSIS cell line expressing autoantibody BCR with the receiverase, and we find the growth of NAPSIS cell expressing 1E8 BCR was dramatically abolished by interferon gamma CAR T cells, and the concurrent interferon gamma CAR T cell had the potential to eliminate cells with autoantibody BCR in vivo. We further investigate the safety of interferon gamma CAR T cell. The cross-reactivity of interferon gamma receptor could allow the CAR T cell to kill receptor-expressing cell. To answer this question, we engrafted two leukemia cell lines into separate positions of NSG mice and assessed the receptor cross-reactive toxicity. So in this animal model, we find the CD19 and all the interferon gamma CAR T cell preferentially reside in the tumor expressing 1E8 BCR and the CD19. And we further assess the T cell activation marker in the TILs from respective tumor. So compared to the control, we can see the CD19 and interferon gamma CAR T cell show much higher and significant level of activation marker in the autoantibody BCR expressing tumor, and there's no difference of T cell activation from the interferon gamma receptor expressing tumor cells. And this result indicate interferon gamma CAR T cell do not respond to leukemia cell with interferon gamma receptor. To further validate the efficacy of interferon gamma CAR T cell on eliminating autoreactive B cells from patients with autoantibodies, we generate interferon gamma CAR T cell from the patient-derived T cell and concurrently the patient with and the co-culture the patient PBMC that differentiate into antibody producing B cell. As you can see, compare the control CAR. There's no detectable anti-interferon gamma autoantibodies, and this result suggests that autoantibody producing B cell will eliminate through interferon gamma CAR T cells. And let's study and taken together based on our finding from the fully monoclonal antibody against interferon gamma, we can develop a unique autoantigen to recognize by autoantibody BCR but not the interferon gamma receptor. And this CAR T cell can specifically deplete autoreactive B cell without off-target toxicity. And this could provide the principle of concept for therapeutic application on autoantibodies related disease. And last study, I would like to thank my mentor, Professor Jordan Gu, for my support. And the CAR T project is performed by my talent colleague, Zhang Jiepeng. And I would like to thank our collaborator and our clinical physician and our funding agency. and I would like to happy to take any question. Thank you.
So thank you so much, Hangpo. We'll ask the question at the end of the talks, of all talks, but once again, I'm encouraging you to ask your question in the Q&A. So our second speaker is Dr. Paul Basta, an MD PhD currently working as the fellow in the Pediatric Hematology and Immunology Department of the Necker Hospital for Sick Children in Paris, and is also doing research in the lab of human genetics of infectious disease, both at the Imagine Institute in Paris and at the Rockefeller University. So Paul has a fellowship supported by the Bettencourt Foundation and INSERM, and his research focuses on the genetic and immunological determinant of severe viral disease in humans, including the cause and consequences of autoantibodies against type 1 interferon. And Dr. Bastard will make this overview on autoantibodies against type 1 interferon and present some of our recent discovery. Thank you, Paul.
Thank you very much, Anne. I hope you can hear me well. So thank you. We share offices with Anne, so I'm very happy to be here. I hope I won't repeat what some of you might have already seen about autoantibodies against type 1 interferons. It's an overview, really brief, of our work and the work of others on the topic. No conflict of interest. And as the usual introduction, the question we asked is, why do people die of viral infections? This is an image from an ICU in England. And so in the lab of Jean-Laurent Casanova, we recruited many patients, and many collaborators around the world helped for this recruitment. And I thank them again here. Patients with asymptomatic or, by contrast, very severe COVID-19 pneumonia. We focused for many reasons on type 1 interferons, which are key antiviral factors, because they were discovered to interfere, and they were the first interferons discovered with viral growth. And importantly, genetic defects in the type 1 interferon pathway are known to underlie severe viral diseases. And early on in the COVID pandemic, we identified several patients with life-threatening COVID and severe viral infections. And so as for interferon gamma autoantibodies, which were brilliantly presented a few minutes ago, we hypothesized that patients with autoantibodies against type 1 interferons, alpha, omega, or beta, for example, could have severe viral disease because of these autoantibodies. Now, interestingly, there was already one report, just to show that we never invent, we perhaps reinvent, of one patient with severe VZV disease because of autoantibodies against type 1 interferons, and this was described as early as the 1980s. So the idea is kind of counterintuitive, but it's a topic of this whole meeting, is autoimmunity against immunity. And we like the image of the snake. This is why I show it here. It's a snake from Greek mythology. So in COVID, we found that 10% of patients with these autoantibodies, both in the adult and pediatric cohorts, in the most severe cases, had high titers of autoantibodies neutralizing interferon alpha and or interferon omega, and sometimes both. What was important is that they were found before infection in all cases tested, sort of proving that they were pre-existing infection. Now, Charlie Rice's team at Rockefeller University showed that they were able to block the protective effect of interferon alpha-2 against SARS-CoV-2 replication, and that even when we dilute the plasma of these patients with autoantibodies 100, 1,000, 10,000 times, they can still lead to high viral replication, perhaps even more than a monoclonal antibody that we bought commercially. Now, the major risk factors of dying of COVID-19 is being an old person. And so we found that these autoantibodies were indeed more frequent in the elderly, and also slightly more frequent in men, which is another risk factor. And so we wondered about other viral infections, and first studied a very severe disease that is extremely rare, which is yellow fever live attenuated vaccine adverse events. This was in 2021, where we studied a small group of patients, 8 patients in total, who had either encephalitis or sort of a disseminated disease following yellow fever vaccination. What was interesting is that when we went back into the old literature, looking at the risk factors of having such adverse reaction to this live vaccine, we found that they were the same risk factors of having autoantibodies against type 1 interferons, being an elderly man, having lupus, or having a thymoma. And so we found that 30% of these individuals had high titers of autoantibodies against interferon alpha 2 and interferon omega. And again, we showed that with Charlie Rice's team, that these autoantibodies could block the protective effect of interferon alpha against the vaccine strain of yellow fever, leading to extremely high viral replication, even at high plasma dilutions. So these events are extremely rare. And we next, and this is the work of Adrien, a PhD student in our lab, looked at a more frequent viral disease, which is also a flavivirus, just like yellow fever. And so we don't see it so much in Paris, but perhaps the colleagues in Italy and Eastern Europe see it much more. It can actually be very severe. In 1% of cases, patients have encephalitis, and the mortality in these cases is really high, 5 to 20%. So we studied a large cohort of 600 individuals with adverse severe encephalitis or mild forms. These are the green patients, or benign forms with asymptomatic infection, the black dots here, 600 individuals in total, and looked for autoantibodies blocking type 1 interferons. So here the figure is inverted. It's a neutralization assay. So if the patients don't have autoantibodies, we see a high induction following interferon alpha stimulation, alpha 2, beta, or omega. And so none of the asymptomatic individuals have neutralizing autoantibodies. By contrast, and extremely notably, more than 40% of patients with the encephalitis form had neutralizing autoantibodies against usually interferon alpha 2 and or interferon omega, and rarely interferon beta. Now, because this is a neurological disease, we also looked in the cerebrospinal fluid samples that we had for a few patients and found that the autoantibodies were almost always found at similar titers in the CSF of these individuals, sort of really showing that they're pathogenic in the site of disease. Now, I won't go one by one to all the different viral diseases that can be in part explained by these autoantibodies, but both the genetic defects and their autoimmune phenocopies affecting type 1 interferons with autoantibodies neutralizing alpha 2, omega, or beta rarely can lead to severe VZV disease, to skin and disease following HSV infection, especially in APS1 patients. I've discussed about COVID pneumonia, but also patients who have been double vaccinated and have breakthrough COVID pneumonia. We found 5% of patients with severe influenza who had these autoantibodies, about a third of patients with the MERS infection in the Middle Eastern. And then Adrien and Shenying Chang in the lab really worked on the flaviviruses. And in addition to yellow fever and West Nile, we found a significant proportion of cases, of severe cases of patients with tick-borne encephalitis, Uzutu virus, or Ross River virus who harbor these autoantibodies. So now we're looking for more, and I hope to update you soon about the new infectious diseases caused by these autoantibodies. And very briefly, before I finish, one could wonder, could they have a beneficial effect? So this has been known since the 1980s, but the team of internal medicine doctors confirmed this recently and found that in patients with lupus who harbor in about 10% of cases autoantibodies neutralizing type 1 interferons, the patients in red here who have the autoantibodies have no flares, so lower disease activity. And this is interesting because a new treatment for lupus is actually blocking the type 1 interferon receptor with anifrolumab. So these patients sort of auto-treated themselves. And by contrast, they have a higher risk of severe disease, either zoster or severe COVID-19. And knowing that these autoantibodies pre-existed infection, we wondered about the general population. And here, both in the pediatric cases and the adult cases, we realized that these autoantibodies are rare below 65 years of age, but rise after 65 years, reaching really high proportions, 4 to 5% of these healthy individuals who are nevertheless at high risk of suffering from very severe disease. And now we're working with a team in Necker to try to find the B cells producing these autoantibodies and prove that they really pre-existed COVID. And to finish, as we know that they pre-existed COVID-19 and other viral diseases, we're now looking for their causes. And so what is known about this? Well, the first known causes of autoantibodies against type 1 interferons is a high interferon exposure, maybe in specific individuals. This has been known since the 1980s, again, in patients treated with recombinant interferon for hepatitis C or multiple sclerosis. I discussed about lupus patients. It's also known in adults or elderly patients who suffer from thymoma and who have a degree of thymic dysplasia. And so this sort of led us to look at inborn errors of immunity in genes affecting specifically the thymus. And the classical example for this is patients with autosomal recessive or dominant deficiencies in the AIR gene leading to APS1, who have multiple autoantibodies against endocrine organs, but who all have autoantibodies against type 1 interferons. And Tom Levoyer in our lab recently discovered that patients who have an NF-kappa B alternative pathway deficiency have an impaired AIR expression in thymus and autoantibodies, again showing that the thymus is really important. But the thymus really works together with T cells, and so there are several groups who described patients with T cell defects, the more classical one being RAG1 or WAG2 hypermorphic deficiencies, who suffered from very severe cases of infection, and these patients had autoantibodies against type 1 interferons. So now we're looking for new novel genes, either important on the thymic side or on the T cell side. With that, I mean, all my other colleagues are going to discuss all these other autoantibodies, autoimmunity against immunity, which are absolutely fascinating. For the type 1 interferon autoantibodies, it's potentially a major cause of death because of COVID and other viral diseases, millions of people potentially. It's really easy to diagnose, and we're happy to share our tests with you if you want to set it up. There are many questions regarding treatment, and I think that Professor Hale will discuss some of them later. And now we're screening more viral diseases and trying to find the genetic causes underlying these autoantibodies, either germline or somatic. With that, I'd like to thank Anne very much for the invitation. All the colleagues around the world who recruited all the patients and helped us with these studies, all the people in blue here, we did all the experiments with them and we still continue, and thank you, and the clinical team. Here are a few pictures of all the team, and I'd be happy to take questions at the end. Thank you.
So thank you so much, Paul, for this wonderful presentation. Our third speaker, Professor Benjamin Hale, has a PhD in molecular virology. So he founded his independent research group at the MRC University of Glasgow Centre for Virus Research in 2011 in Scotland. Then he moved to Switzerland, to the University of Zurich, where he now serves as an associate professor of medical virology. So Professor Hale research focuses on the molecular biology of human interferon system in viral infection, studying both its normal and disease-related mechanisms. So his team has recently launched a clinical program to study the development and long-term effect of human autoantibodies against type 1 interferons and develop new methods to counteract their harmful effect. So Professor Hale will be presenting on the lifelong persistence of these neutralizing autoantibodies against type 1 interferon and the development of a targeted therapy. Ben, the floor is yours. Thank you. Please do not hesitate to ask questions.
Okay, thank you very much, Anne, for that kind introduction. And thank you also to the preceding speakers for their very nice talks. We certainly have been inspired by the work that Paul described on anti-interferon type 1 autoantibodies. So for the past couple of years, my lab has been interested in the question also of what is perhaps triggering these autoantibodies in individuals, particularly in elderly individuals. And also we've started to think a lot about how we can come up with some new therapeutic strategies to counteract them. So this is very much from a molecular biology perspective. So just as a disclaimer on the second part of my talk, the University of Zurich has a patent application related to inert interferon 1 type family members. So this may be seen as a conflict of interest. So we were certainly inspired by the work that Paul described, that these anti-interferon autoantibodies have a higher prevalence in the elderly population. And we have access to a longitudinal study of individuals who've are living with HIV in Switzerland. So around 20,000 people are involved in this cohort. And so we were interested to screen these individuals, certainly those over the age of 65, to see which of them had anti-interferon autoantibodies. Because we also had access to historic plasma samples from these individuals. So these individuals have kindly donated plasma samples every 6 months for the past 35 years. And so we were interested to identify individuals who had these anti-interferon autoantibodies and then work backwards in time to try to work out when they developed these autoantibodies, and if we could understand from their clinical records and their biological samples factors associated with development of these autoantibodies. So consistent with what Paul has shown in this cohort, which is a set of individuals living with HIV, although they're extremely well treated and have a life expectancy similar to the general population. Similar to the general population, we identified these autoantibodies in around 1 to 2% of individuals over the age of 65. So here's just one example of an individual, because we can look at their plasma samples over time. So we can see when these autoantibodies develop, they're negative from the age of 40 onwards when we have the first sample, up around to mid-50s or early 60s. And then they go positive for autoantibodies, in this instance against interferon alpha, and then later against interferon omega. We can also look in these plasma samples and see if these samples are neutralizing, so they're able to neutralize high levels of interferons also. So I think from this set of data we can also conclude a few things. So again, the median appearance of these autoantibodies was around the mid-60s for the individuals that we were able to test. What we noticed was that when these autoantibodies develop, they're not transient in nature. They basically persist lifelong in the individuals, so at least for 15 years. And they always seem to increase in titer over time as well. We're able to look at the PBMC levels or ISG levels in PBMCs post-development of these autoantibodies compared to ISG levels before the individuals develop these autoantibodies. And what we can clearly see in 13 patients seen here is a general loss of ISG levels. So baseline levels of ISGs in PBMCs are reduced following development of these autoantibodies. And then we can also show that these autoantibodies predate subsequent severe infections. In this instance, 20% of the individuals we followed developed severe COVID-19 requiring hospitalization, and they developed these autoantibodies around 2008 or 2010, so 12 or so years before COVID emerged. So these really did predate these severe infections. We're also able to look at some factors associated with development of these autoantibodies, and as Paul had intimated, some kind of T-cell defect or loss of tolerance predates development of these autoantibodies in these individuals. And also we have some anecdotal evidence that around the time of developing these autoantibodies, they have a high interferon status, perhaps through some kind of severe infection. So the question arises, how does one mitigate these pathogenic effects of these anti-interferon autoantibodies? And as has already been talked about, using perhaps treatment with an interferon that is not targeted by these autoantibodies could be a very good therapeutic strategy. In this instance, for example, interferon lambda or interferon beta, if the antibodies are against alpha and or omega. However, there are some issues with this. There is the possibility, if interferon itself is associated with developing these autoantibodies, of raising additional autoantibodies against the treated interferon. And of course, giving excess immunostimulatory type 1 interferon can have pro-inflammatory and potentially negative consequences. So here's just a case to demonstrate that, actually a case from Basel, some clinicians that we've been working with. This individual had a chronic encephalitis, meningoencephalitis, potentially caused by a viral infection, although a virus was never identified. In fact, the individual suffered with this kind of set of inflammatory lesions for almost 10 months. The only factor that we could identify in this individual was that they had anti-interferon omega IgG that was neutralizing. So part of the treatment regimen was to give this individual interferon beta, which was able to, or at least correlated very well with a remission of all of the symptoms of this encephalitis within about a month. But unfortunately, we've been tracking this patient now for many months, and following treatment with interferon, this individual did develop anti-interferon beta autoantibodies. Fortunately, they're non-neutralizing, so we're interested to understand why that's the case. But it just demonstrates that treatment with interferon beta in someone who's prone perhaps to making anti-interferon autoantibodies could lead to development of new pathogenic autoantibodies. So we've been starting to think about new strategies that we could use to mitigate the effect of anti-interferon autoantibodies. So recently we've taken the approach to try to map footprints that these autoantibodies that individuals have recognize on type 1 interferons, with the idea that we could potentially develop molecules that can act as decoys to bind these autoantibodies and stop them from affecting endogenous interferon, at least in cell culture as proof of concept type therapies. So one of the assays that we've been exploring to do this has been taking interferon, interferon alpha in this instance, and tagging it with a small tag, 11 amino acid hibit tag, that we can then quantify using a nanoglow reagent, so we can complement this hibit to generate full-length luciferase. Therefore, we can quantify the abundance of this interferon that we can produce in mammalian cells. And we can combine this with immunoprecipitation type assays, where we can precipitate equal amounts or use equal amounts of input interferons, precipitate this with an antibody against interferon alpha and detect the amount of precipitated interferon. This is specific, it doesn't get precipitated by an interferon omega antibody. And you see the same thing if you use plasma samples from a patient who's positive for these anti-interferon autoantibodies compared to a patient who's negative for these autoantibodies. So using this kind of assay and then a panel of interferon mutants that we generated in the lab, this assay also using Western blot based assays as well, what we were able to do is study around 20 different donors, so aged individuals over 65 who had these anti-interferon autoantibodies against interferon alpha, or a set of 10 that suffered from severe COVID and had anti-interferon alpha autoantibodies. And generally, all I want you to take from this figure here is that the reactivity to the panel of interferon mutants that we tested was similar for all of the patients that we tested. So these are polyclonal plasmas. We didn't clone any antibodies for this, but blue color means very high reactivity to the wild type interferon, and anywhere where there's a white color, you see a loss of reactivity in one of these types of assays. What you can see is that all of the patients had very similar patterns and loss of reactivity. So just to then highlight what some of these residues are on interferon, on the left here is interferon alpha 2 in purple, and we've highlighted in green the residues that we found to be very important across all of these 20 individuals for binding to these interferon autoantibodies. This is shown here in green. On the right, you can see now the binding site for the interferon alpha receptor 1 subunit. So you can see that this green overlaps with the orange quite nicely. And we also identified a second site that seems to be an important site for these autoantibodies binding, which is on the opposite face of interferon alpha 2. So basically the site that overlaps with the interferon alpha receptor 2 binding residues, which are shown here in orange on the right. So there are two faces on interferon alpha, at least two faces that we've identified where there are common residues that are important for binding to autoantibodies from these two diverse cohorts that we've tested. And in data that I can't show you today, just for time considerations, we can also show that these patient plasmas are able to block the interaction between interferon alpha and both receptor 1 and also receptor 2. So that's part of the neutralizing mechanism. But what we wanted to do was take this knowledge of the footprints recognized by these autoantibodies and try to come up with a decoy molecule that could be bound by these autoantibodies. Ideally, this would have the structure of interferon, so would look like interferon to these autoantibodies, but of course be completely inert and unable to activate signaling. So very similar to what was described in the first talk by Anne Puel for interferon gamma. Oops. We thought that, of course, the best starting structure to maintain the important structures would be interferon alpha itself. What we found was that even though these autoantibody footprints overlap somewhat with the receptor binding sites of interferon alpha, there are some unique residues on interferon alpha that are not targeted by these autoantibodies to a great extent. These are shown here in orange, but they are required for binding to the receptor. So what we did was we performed a screen where we mutated one by one these orange residues that are unique for contacting the interferon alpha receptor but are not seen to be required for binding to the autoantibodies, and we looked for loss of function in stimulating cells and inducing antiviral activity. Two residues popped up to us, one on the interferon alpha receptor 1 binding site and one on the interferon alpha receptor 2 binding site, highlighted here in blue. But individually, as a single amino acid changes, these still retain some kind of activity when you put very high amounts of these interferons onto cells. So these are not ideal and are not completely inert. So what we did was we combined these residues into a double mutant as shown here, and this double mutant then is completely inactive and inert. So it doesn't matter how much interferon or this mutant interferon that we put onto cells as shown here, it's unable to stimulate a reporter. What we could show is that this, what we call a signaling inert mutant interferon alpha containing these two amino acid changes is still able to be bound by autoantibody containing plasmas to essentially exactly the same levels as wild type interferon alpha 2. So we're not losing any kind of reactivity whatsoever. And then we can test whether this SIM interferon alpha is a potential decoy by mixing it together with donor plasma containing anti-interferon autoantibodies or mixing a control protein with these anti-interferon autoantibodies from plasma, and then coming in and adding exogenous interferon and seeing if this exogenous interferon is still inhibited by these donor plasmas in this context of this potential decoy molecule. So what you can see here in gray is if you add increasing amounts of plasma containing autoantibodies, you see a reduction in the ability of interferon to signal in this reporter assay that we've shown here. But if we've already mixed these patient plasmas with this inert interferon alpha that does not signal at all, you can see that this inhibitory effect is blocked, and this is common for multiple different patient plasmas. So it looks like this mutant interferon can act as a decoy molecule and block the action of these autoantibodies. We can also show that this has a similar effect on virus replication. So if we essentially do the same experiment where we add the mutated interferon to this plasma autoantibodies and then mix that with exogenous interferon, and we test whether this exogenous interferon still has antiviral activity. So here we use an H5N1 influenza virus that encodes GFP In the absence of any treatment, the cells go nicely green, but if you add interferon alpha, then you can inhibit virus replication, and this is blocked if you have this donor plasma that contains anti-interferon autoantibodies, as was shown previously by Paul. If we mix in the control protein, then we see no effect of this control protein on mediating or blocking the effect of this patient plasma, but we can block that with this SIM interferon alpha 2, this decoy. So this molecule is able to bind to the patient plasma and stop the patient plasma from inhibiting the interferon that we've added. So the interferon again has antiviral activity to block virus replication. So just to summarize some of the things that I've been talking about, what we'd found in our longitudinal cohort from individuals over 35 years was that factors such as loss of tolerance combined with a high interferon level can cause lifelong persistence of autoantibodies that can have pathogenic effect decades later. Some therapies, or the only therapies that are really available at the moment, is to give exogenous interferons that are not targeted by autoantibodies, and some of these are approved for other treatments like multiple sclerosis. But as I showed you, they can have caveats of potentially inducing new pathogenic autoantibodies. What we did was try to map the regions on interferon that are bound by plasma autoantibodies to show how they block the interaction with the receptors. This allowed us to make some mutations in interferon to make a signaling inert mutant interferon that potentially has can be used as a decoy strategy to prevent autoantibodies from functioning. So we believe that these SIM interferons could have potentials as decoy molecules, but also they could be used as depletion reagents in something similar to plasmapheresis to remove autoantibodies, because if they fall off, they don't have any stimulatory activity, or very similar to what was mentioned in the first talk, these inert interferons could be used as targeting strategies to go and kill B cells that have the B cell receptor, because these molecules can't bind to the interferon receptor. So all of this work related to these decoy molecules was done by a postdoc in my lab, Kevin Gruen, and the paper describing this actually went online today, so you can read about it in more detail if anyone's interested. Thank you very much.
So thank you so much, Ben, for this wonderful presentation. And just for the last and final presentation, it will be delivered by two distinguished presenters. So first presenter, Professor Sophie Hambleton, who is an honorary consultant in pediatric immunology at Castle Great North Children's Hospital and a professor at Newcastle University in UK with an outstanding track record. So renewed for her research on cellular and molecular basis of inborn error of immunity, Professor Hambleton has made significant contribution to the understanding of inborn errors of immunity, especially inborn error underlying severe viral diseases. So consultant since 2008, her combined clinical and lab expertise has enabled her to effectively manage many complex immune disorders in children. Nationally and internationally recognized, Professor Hambleton was elected to the Academy of Medical Science in 2020 in UK and leads key genomic and quality improvement initiative in immunodeficiency. And our final speaker, Holm, who is a professor in pediatric gastroenterology, director of the Center for Human Genetics, University of Oxford in the UK. So Holm is a leading expert in gastroenterology and immunology, and his work focuses on the intersection of immune regulation and inflammatory bowel disease, and particularly in trying to understand the genetic and the molecular pathway which influence diverse conditions such as Crohn's disease or ulcerative colitis. So his research aims to uncover how the immune system disruption causes inflammation, with a goal, ultimate goal, of developing a targeted therapy that should improve the outcome of patients with such chronic disease, sorry. So Sophie and Holm, the stage is yours to present your work on autoantibodies against IL-10. Thank you.
Thank you very much indeed, Anne, for the kind introduction and for the opportunity to present at this grand round. So Holm and I will do a double act. And instead of thinking about an autoantibody that neutralizes a pro-inflammatory cytokine leading to susceptibility to infection, we'll rather shift focus to thinking about the neutralization of an immunoregulatory cytokine in IL-10, leading to immune dysregulation in the gut. And so in the first part, I will recap the findings that we reported via the New England Journal last year. And for the second part, I'll hand over to Holm to update you on findings since that was published, and to think about some further implications. So the first child that we studied was referred to us from Belfast, and she was then in the second year of life, but her history actually went back to 3 months of age, when she developed bloody diarrhea and vomiting that were ascribed to cow's milk protein intolerance, and which more or less settled when she and her mother avoided dairy. But from 17 months of age, the symptoms returned with a vengeance, and she was losing weight with a lot of pain, hypoalbuminemia, and general evidence of systemic unwellness. A trial of enteral nutrition failed, and so she ended up on parenteral nutrition in hospital, with the endoscopic appearances that you see on the left of your screen. So these were deep ulcers throughout the colon, accompanied by the gross architectural distortion and inflammatory infiltration that you see in histology on the right. So there was no appearance of granuloma within this tissue. So the severity as well as the timing of her presentation suggested that this could be an inborn error. And this audience will be very familiar, well, probably actually with this specific paper of Holm's, mapping out the taxonomy of inborn errors with respect to monogenic inflammatory bowel disease. And at the top here, you see some intrinsic defects within the epithelia of the gut. But in fact, the majority of these defects are within the immune system, both within phagocytic cells, which mediate inflammatory signals arising from the many pathogens and commensal organisms in the lumen of the gut, and the adaptive immune system, where regulatory T cells perform a particularly important function. So perhaps it's not a surprise that the gut is on the front line, protecting us against the external environment, that these inflammatory disorders predominate in the etiology of IBD. With regard to the presentation, I've described to you a very early onset inflammation of the lower gut with ulcerating and actually often fistulizing disease. Disorders of IL-10 signaling have been described, both of IL-10 itself and of its receptor subunits, IL-10RA and RB. So while this child was being worked up from an immunologic and genetic point of view, in parallel, she underwent functional testing of the IL-10 signaling pathway in the lab of Rainer Doffinger, whose name's come up several times today already, because he specialized in establishing these fairly robust in vitro assays. And here, in gray, you see that while TNF can be induced by LPS in the whole blood of normal individuals and then suppressed by IL-10, in our patient here, indicated by the red triangle, there was a failure of IL-10 to suppress TNF production in this whole blood assay. But very provocatively, when the patient's blood cells were washed, taken out of the patient's own serum, and then challenged with LPS plus or minus IL-10, they proved themselves perfectly able to respond to IL-10. So ruling out an IL-10 receptor problem and instead suggesting that a factor present in the serum was interfering with the ability of IL-10 to engage its receptor. Subsequently, we saw a very similar pattern indicated by the red circles on those same graphs in serum from a slightly older child who had presented aged 4, again with bloody diarrhea, abdominal pain and growth faltering, and again with active colitis and the histologic features you see there. And in that child, we had enough serum to do a third-party assay and show that the serum from the patient could inhibit the IL-10 response of third-party PBMCs. So this had the characteristics of an autoantibody-driven process. And so Reiner set up a particle-based immunoassay for such autoantibodies, and this proved to be IgG present in high titer against IL-10, and sort of reinforcing the results that we'd seen with the LPS-TNF assay. This serum was capable of inhibiting a reporter cell line from responding to IL-10. So our formulation in real time was that this neutralizing anti-IL-10 was contributing to the symptoms in our patient. Indeed, she had no other immunological abnormality that we could discern, and her genetic screen didn't show any known defects of immunoregulation. She didn't have autoantibodies against other cytokines. So we discussed this with the family. Of course, it was a very multidisciplinary team that was caring for the child, who until then was treatment naive because the parents had been very against her receiving corticosteroids. But we reasoned that this had to be challenged, and as well as steroid, she had therapy directed against this humoral component in the form of high-dose IVIG and rituximab. And she had a very gratifying response to this, with rapid improvement in her clinical symptoms, accompanied by gradual resolution of the endoscopic and histologic changes. It wasn't overnight, and in fact, when we first weaned her steroid, she flared, and we introduced anti-TNF. But nonetheless, the arc over time was of resolution of her symptoms in keeping with the reduction in titer of anti-IL-10. Of course, her B cells disappeared and later came back, but interestingly, not so much her capacity to form IgG against vaccine antigens, for example. So just in a little bit more detail, this was the dramatic decline in the titer of her anti-IL-10 and recovery of responsiveness to that cytokine in the whole blood assay. The other patient with the milder disease was treated without anti-B cell directed therapy. And although her titer has declined somewhat over time, she has continued to experience intermittent flares of disease and remains on anti-TNF. So our suggestion is that anti-IL-10 autoantibodies may likely play a pathogenic role in IBD, including some very early onset disease, because they seem to be associated with a phenocopy of inborn errors of IL-10 signaling, and we think this might potentially have wider importance in the choice of therapeutic modality, especially if we were to find these antibodies in patients with common or garden IBD. What we don't know is what is triggering their formation, whether they are primary or secondary to a different inflammatory insult, potentially even extending to pathogens such as EBV, which produce biosimilars of IL-10. And we would dearly love to understand better the natural history of the associated disease and its response to therapies. So I'll hand over to Holm at this point, who will tell us about the evolution of the story.
Thank you so much, Sophie. And after this excellent collaboration with Rainer and with Sophie on initially 2 children, one with infantile onset inflammatory bowel disease and the other one with very early onset IBD, 2 unusual sort of events. One of the obvious questions was, of course, how relevant is this for the broader population of patients with inflammatory bowel disease? And so we have since sort of started a project to screen patients with IBD, Crohn's disease, ulcerative colitis, and IBDU. And we have identified a number of patients with autoantibodies that are neutralizing. And just to give you an exemplar, to indeed show sort of this is an acquired sort of immune problem. This is an 20-year-old who developed Crohn's disease with a very, very severe outcome without having any early signs of intestinal disease, without having severe sort of infection history in the past, without having an immunodeficiency background, and then developing Crohn's disease in the early 20s with severe sort of abdominal symptoms, with the need to undergo multiple admissions, multiple endoscopies, and then starting a history of resections, including colectomy, ileostomy, fistulizing disease formation, having then complications of the surgery, including abscesses, including wound healing problems, and a severe deep penetrating disease on histology. This was associated with difficulty to treat, and having had sort of a problem for 1.5 decades of disease without being properly sort of treated in a tertiary center that would regard itself as having excessive experience with thousands of patients with IBD. So bottom line is patients with anti-IL-10 antibodies can present at later stage as well, can develop a very severe disease background, have a very, very high healthcare utilization, are difficult to treat. Next slide, please. So one of the questions that we had was how persistent sort of are the antibodies? Are they just a trigger of the disease, or are these long-term persistence? And so we used a number of assays to investigate this. This included assays that Sophie had described previously that are sort of binding assays. We used additional assays that are competitive ELISAs, and we used STAT3 reporter assay, a HEC-Blue assay as well. And this HEC-Blue assay, we looked for controls that are shown in green that have very, very strong STAT3 reporter activity and are very high. And then we used patients with autoantibodies against IL-10, where IL-10 exogenous recombinant IL-10 is blocked by the serum. And if this is blocked, then it leads to low STAT3 reporter activity. And then a number of patients, and all lines show individual patients where we had access to samples, typically sort of between 7 and 12 years follow-up. It did show that these autoantibodies are largely persistent and are present even after a decade of disease. So suggesting that these autoantibodies are not just a trigger, but a long-term driver of disease activity. Next slide, please. Next, we wanted to understand sort of some of the functional components of disease. And here we went back to studies that we had done in the lab a few years earlier. Dominic Aschenbrenner and Maria Quaranta in our lab had done some tests to investigate the role of IL-10 signaling in the immune system. They used PBMCs, and in these PBMCs they stimulated with LPS and then added an anti-IL-10 receptor antibody. And a bit to our surprise, we did not only see an increase in inflammatory cytokines, that was expected, but there was a hierarchy of inflammatory cytokines. And the strongest cytokines suppressed by the presence of IL-10 is IL-23, then GM-CSF, then IL-1, then followed by other cytokines, and much less relevance there. So it's only a few cytokines that are significantly regulated by IL-10 signaling. So with this knowledge in hand, we went back to patient cells. Next slide, please. And we used sort of sera from patients with IL-10 autoantibodies, incubated those with PBMCs from a healthy donor and added LPS. And what we were able to see in this in vitro assay that IL-10 was not recognized in those sera who had autoantibodies, presumably because the detection epitope is masked. But in accordance with this hierarchy of inflammatory cytokines, IL-23 had the strongest sort of signal, followed by IL-1, IL-6, and less so TNF. In TNF, I have to say there were a few patients who didn't show any response at all in our cohort, and that was explainable by the infliximab or adalimumab antibodies that these patients received in a therapeutic setting, basically blocking this response as well. So the learning point from this assay was it's probably a bit more functional and physiologic, showing sort of that these antibodies can increase an inflammatory response, but it is more prone to therapeutic intervention as well. So it can be influenced by, for instance, therapeutic antibodies against TNF or IL-23, ustekinumab or P19 antibodies that we are nowadays use in clinical practice. Next slide, please. Taking all of this together, the presentations that Sophie gave and the points that I raised, we have now sort of solid evidence that the blockade of IL-10 either by a knockout in a mouse system or by neutralizing antibodies against IL-10 receptor can induce in a mouse system robust intestinal inflammation. There is very clear evidence that Mendelian disease variants, B allelic loss of function variants in IL-10RA, RB, IL-10 cause infantile onset inflammatory bowel disease. We have some evidence that variants in IL-10RA might have a mild effect, a modifier function in classical forms of Crohn's disease in particular. And the studies just described provide evidence that neutralizing anti-IL-10 autoantibodies can cause a range of immune pathology from infantile onset or adult onset disease. With this, I like to thank the collaborators, in particular Rainer Dofinger from Cambridge, Sophie Hambleton, the team members from Newcastle, Belfast, Cambridge, and Oxford. And the latest studies that I've presented were generated by Nima Garadaki and Charlie Ye in our lab. And of course, many thanks to all the patients who contributed their samples and the clinicians who contributed to this knowledge base. Thank you.
So thank you so much, Hangpo. Then Paul, Benjamin, Sophie, Holm. I think I forgot no one. So for this wonderful presentation. So we have a number of questions, and I guess the first one is almost for all of you. So we are starting to screen patients with susceptibility, with suspected immunodeficiency and having autoantibodies against cytokine. And so the question is, there are more and more individuals found with these autoantibodies against cytokine. So is this increasingly observed elsewhere? So meaning that, do you see an increasing number of individuals with these autoantibodies against cytokines? And the second thing is, what the approach when the results return to the clinician? So do the panel recommend they are followed by functional testing, meaning do you consider that we should test this autoantibody for their neutralizing activity, I guess? So maybe, so I think it's for all of you. Okay, Hangpo, you were the first one. So do you think that, so regarding interferon gamma autoantibodies, so these autoantibodies against interferon gamma, we found them mostly in Southeast Asian patients, but so do you think that when you have a patient with this autoantibody, do you assess them for the neutralizing activity?
So sorry, assess the neutralizing activity.
For all patients, when you detect these autoantibodies, each time do you assess this plasma for the neutralizing activity?
Okay, because we have a recruit about around 200 cases in Taiwan, and in my memory, I think there's almost one or two cases has a non-neutralizing, but all of them are almost all neutralizing, and we have tested for the healthy donor as a control, and surprisingly, we find some among maybe one of the 100% will have a non-neutralizing or even less the non-neutralizing autoantibody for the interferon gamma.
What about you? Regarding the IL-10 or the type 1 interferon autoantibodies?
So maybe for the type 1 interferons, I'd definitely test neutralization. I think it's the most robust and sort of gold standard. I hope you agree, Anne.
I do.
Great.
But we know that there are autoantibodies against cytokine present in healthy individual, and even autoantibodies that stabilize the cytokine. So do you think that some of the disease we will see in some days can be due to antibodies, autoantibodies, not only some neutralizing the cytokine, but some stabilizing the cytokine and making the life longer for this cytokine? Do you see this in your patients? Sophie, Holm, go ahead.
So experience is still growing. You know, anti-IL-10 is the newest kid on the block. So I think we don't yet know exactly what to expect. And in the main, one would err on the side of caution. But I suppose the question depends on the population that's being tested and the sort of pre-test probability a bit of the autoantibody being pathogenic. So if you find very high titer anti-IL-10 in the context of ulcerating inflammatory bowel disease, there's the higher probability that it will be neutralizing. That was all I was going to say.
Yeah, I would agree. Sort of, we need sort of for IL-10, we need better sort of data, more population-based, broader sets. But the important thing at the moment is to look for neutralizing antibodies. So we sort of always combine two assays. One is a competitive assay now, and we add on a neutralizing cellular assay to be absolutely sure this is not just an epiphenomenon, and we measure sort of a binding to a tail that might not be relevant, for instance, for binding to the receptor, because of course the consequences could be sort of as relevant because we don't have guidelines. Some of the treatments, for instance, like B cells of depletion, are not without any harm. So you want to be sure that you are treating the right patient with the highest probability to benefit from these treatments. On the last question, whether it's possible, I'm not aware of sort of that there is a lot of literature, but of course there is enough mouse data, for instance, for some cytokines like IL-2 that typically have a very short half-life, and it seems to work, that there are stabilizing antibodies. I would not be surprised if once there is one human outside somewhere, so who might have stimulating antibodies and might have a degree of a lymphoproliferation syndrome, even with some high Tregs proportions present by a stabilizing sort of factor, for instance. So these things are predictable. It is just how to find some of these patients. There must be a number of diseases out there where we already have the functional evidence.
Thank you. So one question for Ben. Would you expect your SIM interferon alpha decoy to stimulate B cells bearing the relevant O20 reactive receptor? You might that play out.
Yeah, I think that's certainly a possibility, and that's why we shouldn't just think of it as a straight decoy molecule, but also potentially either as a depletion reagent in plasmapheresis or as a targeting molecule, as Han Puel said. But I guess unless that's actually tested, we're never really going to know whether there is a boosting effect and whether this is then problematic or whether the levels are already high enough. So I think that's where I would respond to that. I mean, we just don't know, but it's certainly a concern that one would have to think about and test.
So one for, okay, because there are questions in the two. So one for Paul. Hi, wouldn't anti-interferon type 1 antibody be linked to the thymus and air, also in Oman syndrome? Loss of air expression has been reported? Question mark. Or do you think that T-cell lymphopenia itself can cause these autoantibodies?
That's a very good question. I haven't checked.
Paul,
I do think you need B cells producing these antibodies. So if you refer to Omen syndrome and SCID, oh, yes.
Paul, we
Can you hear
cannot
me?
hear you, so the connection is super bad.
You can't hear me?
So we could, yeah.
Can you hear me now?
Yes, perfect. Go ahead. So restart
Okay.
from the beginning because we couldn't hear you.
Sorry, I hope you can hear me now. So
Yes.
I guess you would, if you refer to Omenn syndrome in SCID patients, I hope I understood well. I guess you would need B cells to produce these autoantibodies. If you have hypomorphic SCID or KID, I guess you could have them. And for T-cell lymphopenia, we haven't checked, to my knowledge. Maybe the others in the panel might know. But it would be interesting to double check.
Thank you. So one is for Benjamin and Paul. Maybe Benjamin, you could take it. So why don't anti-interferon type 1 autoantibodies have a wider impact on antiviral immunity beyond COVID and a few selected viral diseases?
That's probably one better answered by a clinician. I would deflect that to Paul. However, I mean, I would just think of it. I think of the interferon system in a very simplistic way, where it's really important if you don't have other defenses around, like antibody-based defenses. So, then it's really important whether when you're a small child and perhaps you don't have antibody defenses against certain viruses, and then you really need an interferon system to slow down the virus, or when you meet another virus for the first time, like COVID, or some other kind of zoonotic virus like West Nile, and you don't have antibodies already pre-existing against that to protect you. So I think the spectrum so far of viral diseases that have been coming up, COVID, West Nile, maybe also I think flu in that instance, these could be either you don't have other kind of defenses, or the antibody immunity that you do have is waning against that, for example, against that particular influenza strain, and you don't have a humoral form of immunity. That might be a very simplistic way of thinking it, or I'm sure it is a simplistic way of thinking about it, but that's how I like to think about it, at least.
So Paul, do you have additional?
Yeah, I would fully agree with Benjamin, and perhaps, I mean, the more virulent the virus is, I guess you would imagine you need a quick sort of type 1 interferon barrier to fend it off until the adaptive immunity comes in. It's very simplistic too, but I guess it completes.
So one for Sophie and Holm. So P1 in the New England Journal paper was probably symptomatic by age 3 months and certainly by age 18 months. So any evidence for the presence of these autoantibodies in the mother?
Yeah, good question. I wish we had checked that, but we didn't. Partly because by the time we were looking after her, she was sort of well over 18 months, and it didn't really seem plausible that with the level of autoantibody that we were seeing, that it was maternally derived. But in theory, one might expect transmission to be at least possible, and would be very interesting to monitor the offspring of symptomatic women, perhaps with IBD. You could hypothesize that their offspring might be at risk.
So, and I guess following this question, does the panel have concern regarding significant transient neonatal immune suppression following passive transfer of the range of anti-cytokine antibodies used in the mother, having a half-life much longer than anticipated?
Is that with regard to therapeutic antibodies like anti-TNF, for example?
For example, I guess.
Yeah, good point. I'm sure someone is studying that somewhere, but I'm not aware of the literature on that.
Yes, it always sort of needs to be sort of one concern and all, and we just don't have enough data And I think it's probably sort of both sort of these treatment options and these neonatal forms of transmission, like neonatal lupus, neonatal sort of autoantibodies. They will have a certain aspect as long as there is no exposure. The children are probably fine when they're through this window of 3-4 months, and maybe there is an interferon or lack of an interferon signature still present, and these of course might modulate the immune system to a certain degree, but I guess we need much bigger numbers to see these weaker effects or exemplars of life where exposure is happening.
So one question for Hampo. Autoantibodies against type 2 interferons, interferon gamma, are they more common among individuals in certain area, or is it supposed to be similar anywhere?
Okay, for our research, we had to find out this patient because this must come Southeast Asia, and there's a literature report. This patient in China will have suffered from the microbacterial infection. They also have the anti-interferon gamma autoantibodies. And a couple years ago, our lab have collaborated with the doctor from Guangxi in China, and there's another infection called penicillin malaithi. This pathogen, also this patient suffer from this pathogen. They have neutralizing against interferon gamma, and there have several cases was reported in Korea and Thailand in my knowledge, and we have sequencing, because we have a hypothesis, we think the HLA is related to the autoantibody. And we sequencing the HLA, and we find out they have a restriction HLA allele from Thailand, China, Taiwan, where they share the common HLA. So we have a hypothesis maybe HLA will association the autoantibody to interferon gamma about its production.
Thank you. So do you find autoantibodies against type 1 interferon in serum of vaccinated people with SARS-CoV-2 who presented clinical problem, and some of them died of SARS-CoV-2 infection? So I guess this is Paul.
Yeah, thank you. We did look for this in 2021 and found that about a third of patients with breakthrough infections, despite 2 doses of mRNA vaccines, had autoantibodies neutralizing type 1 interferons, despite a good antibody response to the virus, to SARS-CoV-2 following vaccination. So we've never seen any patient who had severe disease despite 3 doses. So I guess if you have a super strong antibody response against the virus, it compensates the type 1 interferon defect, as Benjamin Hale suggested earlier. I hope it.
Sorry, so one is on autoantibodies against IL-6. So maybe I'm reading it, maybe I'll try to respond. So because if a cytokine such as IL-6 has many biological effects, one in the few patients described only present infection and lack of increased CRP and no other alteration that would be related to the blockade of the other function of IL-6. How could we explain this? That's a very good question. This for sure. So all patients we have tested with neutralizing autoantibodies against IL-6 have barely no CRP, and they usually display biogenic bacteria, even though Horst von Bernud in Berlin is also studying such patients and such individual, and in some individual you find these high titers of neutralizing autoantibodies against IL-6, but without apparently any severe disease. Horst, if you hear, please jump in. So it's probably that So for sure, the first patient we described, the titers, these antibodies were strongly neutralizing. So probably either other pathway, other cytokines are taking over. Maybe IL-6 is so there is some IL-6 redundant effect. But it's a good question. It's a good question. And the first patient we detected this autoantibody pretty early at the age of 18 months. But again, not besides severe staphylococcal disease, it didn't display, to my knowledge, any other phenotype that we can see in patients with inborn errors of IL-6 signaling pathway. Holm, do you want to further discuss this?
No, I think that's I can't add on.
And we almost are there any role for non-neutralized antibodies, autoantibodies against cytokine in disease pathogenesis? So in antibodies against type 1 interferon but not neutralizing, against type 2 interferon but not neutralizing, or against IL-10 but not neutralizing. Have you seen any impact of these antibodies? Apparently not.
Not to my knowledge, but they're more complicated to study, I would say.
And maybe the last one. So do you have detected patients with neutralizing autoantibodies against cytokine receptors? IL-10 receptor, type 1 interferon receptor, type 2 interferon receptor?
No.
So not to us, no, but apparently no. So it's a little bit more difficult to assess, I guess. Also, maybe it's some days we will. And
it's foreseeable that this is happening. So in neurology, there are a number of new diseases emerging due to antibodies against extracellular receptors, and this is an emerging field. It would not be surprising if immunology sort of takes up as well.
yes, probably. And the last question. So for Anne Puel, some chronic granulomatous disease patients are on long-term interferon gamma. And do you know if they develop autoantibodies against interferon gamma?
Okay, that's a very good question. But for our case and for cooperation, doctor, we have no record this kind of patient, and we think it might be will develop a neutralizing autoantibody because I think the interferon gamma is produced by a lot of stimulation, so I think it's not quite easy to develop this kind of autoantibodies. Yes.
So thank you. It's 6:38, so maybe we should stop here. I have a number of questions, but I will ask them offline. So thank you to everyone. I hope so. In case I haven't seen your question, please do not hesitate to contact the speakers. And if I miss some question, please, I do apologize. But thank you so much to all of you for attending this session, and thank you for the speakers for these fantastic talks. Yeah, so some people are pretty happy. So thank you again. And there are some announcements. Maria Pia, do you want to make them or?
No, Anne, please go ahead. It's okay for me. And thank you very much for this really informative and very well done, Grand Round. Thanks.
So thank you so much. And once again, don't forget to submit your work in the new Journal of Human Immunity, which is, I guess, is going to be the journal of the field. Thank you so much for attending this session. Thank you to the fantastic speakers and this wonderful session. And join the ESID, become an ESID member. Thank you. Have a nice evening, morning, journey to everyone. Bye-bye.
Bye-bye.
Goodbye.
Thank you.
Thank
Ciao, ciao.